today we're going to talk about the difference first between DNA and RNA, and then we're going to go into protein synthesis. So last week we talked about DNA and DNA replication, and so today we're first going to focus on what is RNA and what's the difference between these two things. So first of all, we remember that DNA stands for deoxyribonucleic acid. Remember, we get the DNA from that part. And the reason it's called deoxyribose is because that's the sugar that's involved in DNA. And so similar to that, RNA, it stands for ribonucleic acid. We get the RNA from this part right here. And so the reason it's called ribonucleic acid is because the sugar involved in RNA is called ribose instead of deoxyribose. And so that's what this stands for. So biggest difference between DNA and RNA comes into play with their nitrogenous bases. So remember, a nitrogenous base is the same thing as a nitrogen base, and those are those four bases involved in the structure. To refresh, the nitrogenous bases in DNA, we started with adenine, which we abbreviated with an A. We have thymine, which we abbreviated with a T. We have cytosine, which was our C, and guanine, which is our G. Okay, and then to refresh or to remember on this, we said that in DNA, the A and the T, the adenine and the thymine, are always the ones that pair together, and the cytosine and guanine are always the ones that pair together. So RNA is very similar. The only difference is in this base right here. RNA does not have thymine, and so RNA, like DNA, has adenine. But instead of thymine, it has a base called uracil. And we abbreviate this with EU. And then, like DNA, it still has cytosine and guanine. So you can see the biggest difference here is that DNA has thymine, whereas RNA has uracil. So in DNA, when adenine and thymine always pair up, RNA, adenine, and uracil always pair up. And then just like DNA, cytosine and guanine pair up, and cytosine and guanine pair up here. So we can always tell if we're looking at DNA or RNA strand because if it has a U in it, it's definitely going to be RNA. If it has a T in it, it's definitely going to be DNA. So an example of a DNA strand, like we've written before, it might be A, T, C, G, T, A, G, C, for example. This could be an example of a DNA strand, whereas an RNA strand, remember, we don't have thymine present, we have uracil, and so this example, or an RNA strand could be like CU, A, C, G, A, U, etc. Okay, so you can always identify this because if you spot U's, if you spot that uracil, we know we're looking at an RNA. If you spot a thymine, we know we're looking at a DNA. Okay, so just to refresh again, last week we talked about DNA replication, whereas if we knew one strand of DNA or one side of DNA, we could predict what the other side would be based on the base pairs in the nitrogenous bases. And so just to refresh really quick, if this was one strand of DNA, we could figure out for DNA replication what the other side would be. And so T's and A's pair together, so this would be A, T, G, T, T, T. Okay, remember A's and T's pair together, C's and G's pair together. This would be A, G, A, G. So this is what we did last week. We did DNA replication. This is how we copy a strand of DNA. We're not doing that anymore. This week we are looking at how does our DNA actually code for our body to make proteins. Instead of looking at this to make a new strand of DNA, what we are looking at this week is RNA. So it's the same type of process, but remember we've got a different nitrogenous base in RNA. So if this is our original DNA strand and we want to translate it to RNA, and it's actually called mRNA because it's the messenger RNA. It's the RNA that's going to go and take this message and then take the message to the ribosomes in the cell, and the ribosomes are what actually where the protein is made. It's called protein synthesis. Synthesis means something is being made. And so this messenger RNA comes to get the message, takes the message away, and then goes to get the proteins made. If we are taking a DNA strand and we are going to make RNA from it, it's called transcription. It's transcribing this message to take it away to go get the proteins made. When we're making RNA, it's called transcription, and so we need to transcribe this. Remember the way that the bases pair up like we went over earlier, but we don't have thymine in RNA, we have uracil. And so if I have a T is going to transcribe to an A, whereas if I have an A, normally if it was DNA replication, I would put a T here. But remember, RNA doesn't have a T, RNA has a U. Okay? C's and G's go together, so cytosine turns into a guanine. Adenine, usually for DNA replication, is a thymine, but remember RNA doesn't have thymine. RNA has uracil, so this is going to be U, U, U. Thymine and adenine, thymine and adenine, cytosine and guanine, thymine and adenine. I've got a G. And then the last one again, an A typically would be a T, but because this is RNA, it's going to be a U. And so this is my mRNA, this is my messenger RNA. It does this in the nucleus, because remember the DNA is found in the nucleus. It does this in the nucleus, transcribes it, takes this message out of the nucleus to the ribosome, where we're then going to make proteins. So before I go into the next step, I do want to introduce what a codon is. So a codon is a 3-letter segment, and so we break up this whole strand into 3-letter segments, or codons, because each codon will then stands for, or it translates into an amino acid. Amino acids are building blocks to proteins. So if we can string together a bunch of different amino acids, we can actually make or synthesize proteins. And there's 20 different amino acids that are essential to building proteins. So, like I said, a codon is a 3-letter segment. And so what we would always do is take our long strand and we separate it or divide it into codons. So every 3 letters, I'm just going to separate it. So you can see here, this strand is made of 4 different codons. Okay, so these are your codons. So what happens, your mRNA takes this message from the nucleus where it read the DNA, it takes it from the nucleus, it goes to the ribosome. The ribosome is the little factory that makes your proteins. And so in the ribosome, we've got tRNA. T stands for transfer RNA. And what that does is it brings these amino acids, it reads this code and it brings the amino acids and starts building that string of amino acids to make proteins. So tRNA, if this is a codon, tRNA actually has an anticodon or an anticodon, and that is the opposite of the codon, okay? The opposite meaning we need to say A's and U's pair together, G's and C's pair together, and so an anticodon, think of it like it's the opposite of the codon. Because it's still RNA, we're still gonna have uracil in this. We're not going to have thymine. The only time you're going to see thymine is if we're talking about actually DNA. Anytime we have RNA, any type of RNA, mRNA, tRNA, we've got uracil in there. So if my codon is AUG, my anticodon would be UAG. C because of the way that those pair up. If my codon is UUU, my anticodon would be A. But what's important here is my tRNA is actually bringing an amino acid with it. So this is actually bringing, attached to it, I'm just going to put a star here, for a little amino acid. And so it's saying, oh, it needs this amino acid. This one is saying it's bringing this amino acid. And these bond together, and so as I continue, I've got this string of amino acids which makes a protein. And so if I were to look at the last two anticodons, AAG would make UUC, and that brings with us a little amino acid, and it strings it together. AGU would bring, or the anticodon would be UCA, and that brings another little amino acid. And so what I'm left with is this string of amino acids, which actually forms a protein. Now it looks like we've done a lot of work here. The great thing is that I actually don't even have to do this last step. I just wanted to show you how proteins are actually made or synthesized. The great thing is we have this chart that I'm going to show you, and the chart tells us what amino acids we're going to get based on our mRNA sequence. And so I don't even have to do this last step. I could look at this chart, because the chart is always based on the mRNA. Don't even have to do the last step. I can look at this chart and say, this codes for this amino acid, this codes for this amino acid. And so this chart will help us figure out the amino acids based on the mRNA. I don't have to do that last step. So now I'm going to show you how to read that chart, and that PDF is provided for you so that we can just take DNA, we can transcribe it to the mRNA, and we can figure out what the amino acids are made. This process right here is called transcription. The process where proteins are being made, or protein synthesis, is called translation. It's taking this and it's translating it into what proteins it makes. Okay, so we've got transcription and translation.